about 2/3 of the way through. I know I've asked you this before, so I'm sorry. It's going to be a repetitive question, but just out of curiosity, how many of you guys have worked in a lab and are familiar with like basic microbiology platforms? One dude that'll admit it.
It depends on what kind of lab you're talking about, man.
Like a
The lab you worked in? Absolutely not.
no, just
How
like
much?
microbiology. Yeah. Okay. Okay, cool. Just trying to get a sense of like how, because with IPAP, with freshman classes in particular, oh, he's dining you out big time. Yeah. Well, I figured maybe George would be like, definitely have lots of experience with this stuff. But it is a very diverse background, set of backgrounds in this room, so it is hard to teach some of this stuff. I mean, we throw around terms like PCR all the time, but how many of you guys have delved into principles and theories, besides Major George, of how PCR works? And even today, it's still going to be pretty superficial. I just want to cover like the bare minimum. Oh, it's okay. Okay. Yeah, just like very superficial level treatment of some of this stuff. But these are the must-knows, just so we have an idea of what Microlab is able to offer us in terms of diagnostic tests. And there's some other ones too that we'll briefly talk about, but these are kind of the staples of what microbiology labs use. So, kind of the outline of today, we'll go through all of these. And in terms of which one of these tests we select, we usually oftentimes use a combination of these things, but it is helpful if we have an idea of what kind of organism we're dealing with, what we expect. The way we would work up a bacterial specimen is going to be very different than we work up a viral specimen for a lot of different reasons, and same with a fungal specimen. So having an idea about whether it's a fungal pathogen, et cetera, is really helpful. If you truly don't know, I was talking with one of you guys about meningitis yesterday. That could have a fungal etiology, a viral etiology, a bacterial etiology. Sometimes you really don't know, and the presentation might be very similar. So then you've got to draw a CSF and submit it for everything, all kinds of testing. But usually, hopefully, you can limit that a little bit more. So type of organism suspected. Urgency of diagnosis, use the same example. When your patient has meningitis, that's one of those urgent types of situations where that person really needs treatment fast. So we might sacrifice more specific or higher sensitivity tests so we can get some sort of answer. Order a rapid antigen test, order a PCR because it's that urgent. And then when we have time, we'll read out the results from a culture or something that's a little bit more reliable. So urgency of diagnosis is important. Culture can take a very long time. Availability of resources. Even in the military, even in the DoD, where we have relatively deep pockets, we still have resource constraints. Some of these tests are prohibitively expensive, so that becomes a consideration. Some of them are also very complex, and they require highly trained personnel to execute them. High complexity testing, you might have the money to buy these instruments, but if you can't afford the highly trained personnel to run them, then what's the point in having them? And then that ties in with the expertise of lab staff. If you don't have competent, qualified lab people, it's just none of this is really worthwhile. So whenever possible, because each one of those methods on this screen is nothing is fail-safe, whenever possible we try to combine approaches, use orthogonal approaches, so that we can kind of fill in whatever gaps are left, whatever disadvantages are intrinsic to a single method, combination testing. And most of these are kind of considered to be traditional methods of identification. However, when we're talking about PCR, which falls into the realm of nucleic acid detection, now we're starting to get towards that trend where we are able to offer more rapid results, more accurate results, PCR and other methods like that, course, again, the trade-off is PCR is expensive, and you can screw it up if you don't have qualified, competent people doing it. But PCR, other technologies like sequencing where you actually like determine the genomic sequence of the organism and identify it that way. MALDI-TOF, which is a form of mass spectrometry that some labs use, super expensive platform, but really impressive technology. All of that, even in DoD MedSense, we're starting to purchase some of that higher end equipment that will give us more rapid results. This is on slide 45. It's an FYI. I don't mean to horrify you too much, but it's an FYI. But it gives you an example of some of the considerations if we have a bacterial specimen, just a bacterial specimen. So this would be totally different for a fungal specimen or a viral specimen. We'll go over this again in slide 45 after we've overviewed some of these techniques, but just very briefly, this little chart up, or this little square in the top left is just explaining what types of plates we'd use, what kind of media we'd select for different types of specimens. So if it's a viral specimen, obviously we're not going to culture it at all. Gram staining. This is when you're looking at the bacteria under a regular bright field microscope. If it's a viral specimen, again, there's no point in gram staining, so that's not done for viral specimens. PCR most certainly is, and some of these other techniques, but yeah, at the end of the day, the point here is just knowing what you're doing, knowing what kind of specimen you have is important for determining these algorithms. A strep PCR? There are a lot of really common PCRs. We have common PCRs for Staph aureus, some for Group B strep. Are you thinking Group B strep or another type?
I just know like I've heard that happen a lot.
It depends. So there are some pretty low, like there are different tiers of PCR. We have like the FilmArray, for example, which is very user-friendly, easy to do. You don't have to do a lot of manipulation. You don't have to do extraction yourself. You basically just load the sample in the cassette and pop it in the instrument, and all of that is done on board. Same with the Cepheid Gene Expert, if you've heard of that. If you want to do rapid testing for Group B strep or MRSA, tuberculosis, and some other assays like that, then you can do those. But most of the PCRs in labs are going to be done by molecular biologists who have to do the extractions themselves and do more like a more advanced interpretation of the results. Okay, so that horrible slide aside, let's start talking about culture a little bit. We've already introduced culture as being important for doing antibiotic or antimicrobial susceptibility testing. But what this gives you is the ability to look and characterize these isolates. So you identify them and you can characterize them in a way that you really can't do with any other methods. We've been culturing specimens for over 100 years. This is pretty common practice. So we're good at doing it. It gives a lot of valuable information. But the drawback is you can't really accelerate bacterial growth rates. They are what they are. You're putting them in the best conditions possible. So we talked about those conditions, the right atmospheric conditions, the right temperatures, you give them the right media types. But they're still only dividing once every 30 minutes or whatever their generation time is. So these are usually going to be an overnight growth requirement. And you can't speed that up. So they are time consuming. Some of them, like Mycobacteria, Nocardia, some other bacteria, take days to grow up, just to the point where you can get visible colonies on a plate. So in terms of other reasons why we would culture bacteria, identification we've talked about, but we can also in some instances quantify the bacteria. UTIs, those are probably the best example of quantifying bacteria with culture. When you have a urinary tract infection, if it's like a clean catch specimen, we expect there to be some E. coli in that specimen. So there's sort of an arbitrary cutoff. Or if it's anything less than 100,000 colony-forming units per mil of urine, then they're probably okay. It's either those are commensals or something else. But quantitatively, if you exceed that threshold of 100,000, then you usually just call that a UTI. It's significant, and you probably have a legit urinary tract infection. And that can also be done with some other fluids that are supposed to be relatively sterile. Like But you can do it with blood. You can quantify organisms in blood as well. The characterization, it's not just the morphology you're at, right? The next slide will show some different examples of morphology. But we can use the metabolic characteristics of bacteria, which is very distinct and unique to each type of bacteria to help identify them. And then we can also look for virulence factors as well. One example is capsule production. We talked about capsule and what its use is for bacteria. You guys remember what it does for bacteria? A couple different things, but for one, exactly, yeah, it helps those bacteria avoid phagocytosis and mask antigens and a couple other things. So if you can see a strain of the same bacteria, like Streptococcus pneumoniae, that is capsulated, that's an indicator that it's probably has that virulence factor. And there are uncapsulated strains of that bacteria as well. And lastly, when we do have those colonies, we can set up antibiotic susceptibility testing right away, which is really super valuable for obvious reasons. The whole process, if you're unfamiliar, is very simply get the specimen from the patient, wound specimen, urine specimen, CSF from a lumbar puncture, whatever the specimen is, blood. You take it down to the lab. The lab workers will inoculate it onto that media if they're doing a bacteriology workup. Overnight growth, and then when you come back, you should have colonies that you can work with from there, do your additional workup at that point. In terms of going back to different types of media, the next slide will talk more about specific types of media, but different bacteria do have different growth requirements, so we select those out very carefully depending on what kind of specimen we're working with. Controlled environment, and then very importantly, sterile conditions. If you have contamination that could occur because of the specimen itself is collected poorly, like a blood specimen that has contaminating saprophytes from the skin, that's undesirable. You don't want that. Or like a urine specimen that's been contaminated. But it can also be contaminated in the lab if the lab people aren't handling that specimen. So it's really, again, important to be clean in the lab and have good sterile technique when you're manipulating these specimens. This is an artificial contrived plate. I can't think of any specimen source that would yield this diverse, like extreme diversity in different types of bacteria, but there's a lot of stuff going on. And the reason why I like it is, again, it gives you an idea of how we can describe different types of bacteria on plates and how morphology is useful. So all of these different characteristics of bacteria are useful, and the people reading those plates the next morning will take notes on these different characteristics. And they usually have a pretty good idea of what is considered to be significant clinically versus not significant, something that can be ignored. So all these things are of interest when we have colonies. With this last one, hemolysis, that's what we're looking at on this slide, hemolysis patterns from bacteria. The production of hemolysins from bacteria is one of those examples of a virulence factor that can be used for informative purposes. If bacteria secrete hemolysins when you plate them on a blood agar plate like this one that actually has like sheep blood in it, those hemolysins will break down the red blood cells in that agar and they'll produce a clear zone around those bacterial colonies. We have 3 different types of hemolysis patterns that we use in the lab to describe them. All of them have some sort of clinical significance. With alpha hemolysis, this is considered to be the partial breakdown of red blood cells in the media. So that usually has like a green, sometimes metallic color to it because of that partial breakdown of blood cells. Beta hemolysis patterns like this one, that means those blood cells have been completely hemolyzed. So lysed, and you get a clear zone of inhibition around those colonies. And then gamma hemolysis is when you have no hemolysis at all. So those blood products are not being broken down. In terms of different types of media that we do have basic nutrient media types like tryptic soy broth and tryptic soy agar. We really don't have anything special in them, but most bacteria like to have things like yeast extracts and peptone in the media. So some of them basically just grow with those requirements, non-fastidious organisms. we talked about fastidious organisms previously, but what does that mean? Picky, requiring special nutrient sources. So if you do have these picky fastidious organisms, things like Neisseria meningitidis, for example, then you would want to plate on something like this sheep blood agar plate in the upper part of the slide, or a chocolate plate right there, which has those factors that are available in red blood cells that bacteria like, already broken down and readily available for them to use. So this chocolate plate, for example, is really good at growing things like Neisseria and Haemophilus influenza and some fastidious bacteria like that. For selective media and differential media, these are oftentimes not exclusive, so we'll use selective and differential media types together, but they're important when we have specimens that are likely to have polymicrobial populations, so lots of different bacteria in them. If you think about the human body and all the possible specimen types, does anything come to mind as a specimen where you would want to be able to differentiate or select for different types of bacteria? Yeah. Yeah, so he said in your gut. So what kind of specimen type would that be? There are, but it's stool, right? So if somebody has a severe diarrheal infection and the clinician orders a stool culture on that patient, you're going to get in that, like in feces, you have a ton of different types of bacteria, right? Most of it's non-pathogenic, most of it's not interesting, it's the non-etiologic cause. So it's really important that a lab is able to offer differential and selective media types in order to grow up pathogens specifically, and not the commensals that you're not interested in. So we're talking about pathogenic types of E. coli, talking about Salmonella, Shigella, Campylobacter, all of those things we need selective media, which would have things like antibiotics and salt that would kill other types of bacteria. And then we need differential media that has some sort of indicator, like a dye, like a pH indicator, something like that, that keys us into the fact that we have our bacteria of interest. Examples of selective media would be like MacConkey agar and mannitol salt agar. These are both also differential media types in their own right. MacConkey is used for gram-negatives. It has salts in there that are generally prohibitive for gram-positives, so grow out gram-negatives. And more importantly, the carbohydrate source is also selective and differential. So it will only have lactose in that media. That lactose is important for bacteria that can ferment lactose, like E. coli. And E. coli they've been living in our guts forever, right? So they're adaptive and able to use lactose as an energy source. And they can because they have the LacZ operon, which we all are familiar with, right? So in the presence of lactose, they can express a LacZ operon and break down lactose so you can get glucose from it. A lot of bacteria don't have that. So that's just kind of tying it in with MacConkey agar. And mannitol salt agar, that would be important for things like Staph aureus that are tolerant for high levels of salt where other bacteria aren't. Transport media is the final category that I think is worth touching upon, especially because that's what is important when it's getting from the patient up in wards down into the lab. So transport media is just specially designed to preserve that organism's viability. If it arrives to the lab dead, are you going to be able to culture it or recover it? Definitely not. So those labs will have specific SOPs. You have to reach the lab in this time period. Otherwise, you're probably going to have non-viable dead bacteria that arrive to the lab. Okay, biochemical tests. I was mentioning that the specific types of metabolism that different bacteria have can be used in such a way as to help identify the types of bacteria they are. So their metabolic capabilities are very important, how they ferment sugar, the enzymes they produce, all of that can be useful. In terms of weaknesses, you still have to grow up the bacteria and watch whether or not they can metabolize these different things or produce different enzymes. So we're talking about more time, usually 24 hours, sometimes 48 hours before you get those results from a biochemical test. However, this too is pretty cheap, or it can be pretty cheap, depending on what platform you're using. And these phenotypic traits that you can see are sometimes not always mirroring what you would see in vivo. So that's another problem. Okay. You can hand read these and interpret the results, or more often than not these days we have automated platforms like the Vitek or the Microscan or the BD Phoenix that can read these plates that you set up. But if you look at an example of this plate right here, you see there's been a gram-negative bacteria that's been loaded onto this plate. This one happens to be, it's a gram-negative plate, and you can see there's like 3-letter codes that identify which of those biochemicals or enzymes are in each one of those levels. You don't have to read this. The instrument itself, you load it onto the instrument, and it'll detect which, like the intensity of these different colors, and it'll give you an indication of what bacteria that is. But you can see, you've got rhamnose and inositol, and urease, and lysine, citrate, a bunch of other sugars on those plates that are used to identify that bacteria. And those can be a much shorter time course, 4 to 6 hours if you're using an automated system like that. Okay. Gram stain, this is one of the oldest methods we have of getting information about bacteria. I'm using it for probably 100 years. It's super fast. As long as you have a colony to work with, you can go straight to a gram stain. Or if you have a sterile specimen like CSF or blood, you could do a gram stain directly from that specimen because there's not going to be there shouldn't be a bunch of bacteria in your blood or in your CSF, right? So sterile specimens, you can go straight to gram stain. If it's a specimen like a respiratory specimen, you'd probably want to culture it first and then take a colony from the culture and do a gram stain. But super rapid, very cheap to do. You get immediately valuable information about whether the bacteria you're looking at is gram-negative or gram-positive. How might that be of immediate value to a provider? Yeah, it immediately starts focusing your attention on what would be most appropriate to prescribe in terms of antibiotics for that patient. So just because it's preliminary and non-definitive doesn't mean it's not valuable for you, especially if it's an emergency or an urgent case. However, we'll see this in a second. When you're looking at these slides, the gram stain slides, you're definitely not going to get species level identification. So you might have seen your 100 millionth Staph aureus, and you might be pretty sure it's Staph aureus. But you can't be 100% sure. Okay, it's just some morphology, microscopic morphology. So it's not super specific. Can never get down to genus or species level. And also, this is not definitive. It's just an early indication of what that patient might have. We've seen this slide before that contrasts gram-positives versus gram-negative cell walls, but we're going to talk a little bit about how gram stains are used to distinguish these two different cell walls. This is a cross-section of a transmission electron micrograph, and you can see here the peptidoglycan layer of gram-positive, how much thicker that is, versus that thin little stripe for gram-negatives. Tiny little stripe right there. So knowing those differences are going to be important for understanding how gram stains actually work. Off to the right, we have a figure or a schematic that should mirror what's on the left, the text on the left. And then a cartoon too of what we just saw in the last slide, showing us a really thick layer of peptidoglycan for gram-positives and a really thin layer of peptidoglycan for the gram-negatives. When we start off with our specimen, we're taking it, let's just say, from a plate. So we have a pure colony from a plate. We will then put it on a microscope slide and then do something to kill the bacteria, either by heat fixing them or ethanol. But those bacteria should be killed before you start staining them. And then you dry that slide completely, usually air dry it. So you start off with this unstained slide with fixed bacteria that are completely dead. And then the first thing you do is add a primary stain, usually crystal violet, which is this big, heavy molecule that will permeate through peptidoglycan and bind to it. With gram-positives, there's a lot of peptidoglycan to bind to for that crystal violet dye. And for I'm sorry, did I say gram-positives? It's not? Okay, good. Yeah, for gram-positives, there's a lot of peptidoglycan to bind to. For gram-negatives, there's not a lot to bind to, but initially that crystal violet will bind to the peptidoglycan layer in there. After that, you'll fix the crystal violet in that peptidoglycan layer with something like Gram's iodine, which is a mordant. And for the gram-positives, that basically locks that dye into that peptidoglycan layer. After that, you will decolorize usually with something like ethanol. And the ethanol will, for at least gram-negatives, that outer membrane usually gets destroyed in that process. But here's where we start to get differential. When you decolorize, that thin layer of stained peptidoglycan gets washed away because that iodine hasn't fixed to it and because it's so thin. So if you were to stop at that step, the gram-negative bacterial cells would be clear and the gram-positives would have that crystal violet appearance, that bluish purple appearance. So then the last step is to counterstain with a lighter stain like safranin or some other stain like that. Safranin is pink stain, so when you counterstain a gram-positive with safranin, it's too faint to actually change the color of those cells, and that crystal violet has already been fixed in that layer of peptidoglycan. So they're going to stay blue-purple, but when you counterstain with safranin for a gram-negative, that outer membrane again has pretty much been destroyed, so that safranin will be able to settle into the peptidoglycan layer. That crystal violet has been removed during the washing step. So as a result, the gram-negative will stain like a pink reddish color. Yes, sir.
And there's 15 steps now. Is it instantaneous with the color changes? You have to wait a bit and then let the wait for the color change.
Yeah, there are. Yeah, it's hard to tell visually if you're on the right track with these. So when you're doing a gram staining protocol, you usually have a timer with you, set timer, especially if you're doing high throughput. There's a timer that has how many seconds you have to wait with a primary stain, how many seconds it takes to fix with Gram's iodine, how long it takes to decolorize, and then counterstain. Yeah. So for example, like if you over decolorized, if you spent 2 minutes decolorizing instead of 30 seconds, what might happen is your gram-positives will start having some of that crystal violet leach out of them, and then you could get an erroneous result. Basically, any of these steps can be screwed up unless you're following that protocol very carefully. It's a time-sensitive thing. And it's also important to rinse in between these steps too, which is why I put on this visual of somebody actually performing a gram stain. Most big labs will have this automated as well. So you have a little pre-programmed gram staining machine, and it has a robotic arm that comes out and grabs the slide, dumps it in each one of these stain tanks or mordant tanks or decolorizing tanks, rinses it all of it. It'll run through the entire protocol, and then that takes out the human error of doing this by hand. Yes, sir?
Is that protocol like less than 30 minutes to an hour for you to get your result, or?
Oh yeah, it's pretty fast. This whole process is super fast. Yeah, I mean, even if you're kind of bumbling along, it shouldn't take longer than 5 minutes to do a gram stain on a single slide. Yeah. Anybody ever done a gram stain? Okay. Anybody really good at doing a gram stain? You can brag. It's okay.
Usually something.
Anybody terrible at them? You're terrible? Okay. I think they are super easy to screw up. It does take a lot of experience. So when you are in the habit of knowing what to look for and what looks right and what looks bad, eventually you train yourself into being able to troubleshoot gram staining. But there is kind of an art to it, and it is easy to screw up. These are some examples of what good, high-quality gram stains look like that you would be able to look at and extract useful information from. In the upper left, we'll start there. It looks almost black, but that's a gram-positive, so it's that purplish blue crystal violet stain. So we've got a gram-positive, and then in terms of the morphology, we've already talked about morphology of bacteria before. What's the morphology in that upper left? Yeah, so gram-positive bacilli, and then you can see what in the middle of these cells, especially like right there, right there. Anybody know?
Nucleic acids.
Spores, yeah. Looks like there's gram-positive rods are starting to sporulate. So we know there's only a couple genera of bacteria that can sporulate. It's probably either going to be Bacillus or it's going to be Clostridium. And that's just based on the gram stain. Got that information just from the gram stain. Bottom left, we've got some pink bacteria. What shape are these guys? Also rods. So we've got gram-negative rods, likely gut bacteria, but not necessarily. Pleomorphic, so the different lengths. Some of them are quite long, some of them are really stubby. In the upper right, we've got some gram-positive cocci, and they're in pairs. And then you can see on the caption right there, it says capsules. Can you guys see the capsules on those cells? Yeah, it looks like little halos around those bacterial cells. Those are capsules. So that would be an indicator that that particular strain of bacteria, probably Streptococcus, is a capsule producer, which has implications for its virulence. And then lastly, in the bottom right, we have some gram-negative cocci in pairs. There aren't a whole lot of gram-negative cocci, and especially not in pairs like this. So this is probably either Neisseria meningitidis or Neisseria gonorrhea. Okay, any questions on the gram staining? For testing purposes, I know a couple of you have already identified yourselves as colorblind. If there are any questions asked about gram stain, and if I show you an image of gram positive or gram negative, in the text I will just write out that this is like a purple or a pink. All I'm asking is that you know that pink corresponds to gram negative and purple or blue corresponds to gram positive. Fair? Okay, so then in terms of microscopy and how we'd actually read these, bright field microscopy, just traditional standard microscopy, is how we read gram stains. And that is the workhorse in terms of microscopy capabilities for most medicines, most clinical labs. But we do use some others that are clinically relevant as well. So just a little bit of an overview on some of these other microscopy methods. First, we've got phase contrast. This is an example of phase contrast in the very top here. This is helpful when you're just looking for, these are unstained cells, primary cell lines, and they're living. So if you're doing a viral culture, for example, these are eukaryotic cells. These are viral cells from African green monkey kidney cells that have been infected with virus. So you can't really see it very clearly, but if you zoom in on your screen, these are the healthy cell lines. And then on the far right, this is after a couple days when they've been completely destroyed by whatever virus was used to infect them. So phase contrast is a nice way of looking at living cells for doing virus culture work. And no need to stain, because these are still living cells. With dark field microscopy, basically, this is useful when you have bacterial cells that can't be stained. They might be gram positive, gram negative. But if you are 200 times longer than you are wide, like a spirochete, which is a spiral shape that we talked about, a gram stain isn't going to be able to be useful. You're too skinny. This is like 0.1 microns in width and super, super long. So to overcome that, we can do dark field microscopy, which is basically when you're shining light outside of that bacterial cell. So everything appears in shadows, everything appears dark except the bacterial cell itself. So it just lights up itself. We don't use that super often, but it is kind of the preferred technique if you're looking at spirochetes like that. Next, we have fluorescence microscopy, which is pretty common, especially in labs that can afford it. You need a fluorescence scope, but it's really helpful for things like tuberculosis. This example is showing Legionella, which is another type of bacteria we'll talk about later. But with fluorescence, you're using an antibody to bind to those bacterial cells, and then that antibody is conjugated to some sort of fluorophore. So when you look at it under a fluorescence microscope, it'll light up. So that's really helpful just to determine presence or absence. But morphology, you're not going to get any information on morphology. But you don't need it because your antibody is telling you what that agent is. just want to know if it's present in the sample or not. And then lastly, these two methods, transmission electron microscopy and scanning electron microscopy, these are really not used in clinical settings at all. They're super expensive. They take up an entire room. So this is more what you would see in a research facility. However, the reason why I put it here is because a lot of images I show in this course are transmission electron micrographs or scanning electron micrographs. So more for educational purposes, so you know what they are and what they do.
Is the fluorescence the same as the ELISA test, where it uses the same antibody to mark it, and then like
The LISA test?
the the
Oh, the ELISA?
ELISA?
No. So we'll talk about ELISAs today too. ELISAs are a type of immunoassay, but fluorescence microscopy is supposed to be a really quick way of looking at a specimen and applying, just putting on those fluorescently labeled antibodies and then taking it over to your scope to see whether or not it's there. Similar principle, they both use antibodies, but ELISAs are a different type of test. So with transmission electron microscopy, real quick, the magnification range is significantly better than bright field by like orders of magnitude. And this, by the way, is probably the low end of that range, 5,000 to 10,000 times normal magnification in the human eye. It gets much more magnified than that. But as you can see from this, you're looking at the planar structures, and you can get information, for example, about like what's going on in the cellular process. This is showing us exocytosis going on from some sort of infected cell. You can see, I think it's a time-lapse experiment, but you can see this particle about ready to leave this infected cell. Scanning electron microscopy differs in that you're offered a three-dimensional representation of what you're looking at. And that too can offer a lot of really good information that you just can't quite describe with other normal pictures. We were talking about transduction the other day and how you have transducing phages infecting bacterial cells. Well, there you go. This is a picture of a phage at whatever magnification this is, but a teeny tiny bacteriophage zoomed in, and look at the detail that you can see on that thing. And on the right, you can see zoomed out a little bit, but a bunch of phages sitting on some infected bacterial cell. A lot of detail. Okay, I'm talking about bright field microscopy. This is a very humble, cheap type of microscopy, but we love it because, you know, we use it for everything. Just to kind of orient you to some of the features there. This is the stage of the microscope where you put your microscope slide. We have a light source at the bottom there and a condenser diaphragm just underneath the stage. Fine focusing knob and a coarse focusing knob. And then in terms of the magnification, you're not going to get a whole lot from these microscopes, but you've got different objectives here that if you rotate these objectives, you can change the level of magnification. On the lower end, usually you get like a 10x objective for magnification. Next up is usually 40x. And then the very highest that you can get with a bright field microscope is about 100x. So this is oil immersion, where you'd have to literally place a drop of oil on your slide with a gram stain or something like that, and then lower that stage so the objective is actually touching that oil. Oil has about the same refractive index as glass, so you eliminate some of the distortions and get higher resolution if you put that oil drop on there. So 100x oil immersion is the highest power you can get on that. We'll see in the next slide that still isn't a huge amount of detail that you can get from these specimens. One other thing with the ocular lenses themselves, these offer an additional boost of a 10x magnification. So when you're talking about total magnification of your specimen, whatever that objective is, you multiply that by 10, and that's your total magnification. Okay, so resolving power is not great. The limit is about 200 nanometers, so not super good. On that low power setting, that 10x setting I was talking about, we have not a huge amount of resolution. You cannot get much information at all about the bacteria in a specimen. This is more just for scanning the specimen type. Usually very low level of light. You can see it increases when you bump up in magnification. If you go up to 40x on a regular clinical specimen where you're looking for bacteria, like gram stained bacteria, you may probably start to see bacteria, especially if they're bigger bacteria like Bacillus anthracis. So So here you can see there's some gram-positive rods. You don't see a lot of detail though. So if you bump up to oil immersion on a 100x, using that 100x objective, now you can definitely see gram-positive rods on that specimen. And then we have some other stains we'll talk about briefly. There are many, many, many other stains we won't talk about at all, but these are useful and good to know about. Some of you may have already done this kind of microscopy, but wet mounts are very frequently done, very useful. For example, a very common type of wet mount is when you take like a vaginal specimen from somebody, a vaginal smear, put it on a slide. If you suspect that patient has vaginosis caused by Trichomonas vaginalis, you do it, you do this, and as you can see, this organism is still alive, so you haven't fixed it, you haven't heat killed it or killed it with ethanol, and you can observe on a positive specimen that unique distinctive motility that Trichomonas vaginalis has. So I've seen a couple of those in clinical labs. Another very common type of microscopy specimen that we work with is if we suspect that there are yeasts or fungal like molds in the specimen. So we can do a KOH wet prep. With KOH, that's potassium hydroxide. It degrades most of the cellular tissue and debris in that specimen so that we can have a better chance of looking at the fungal structures there. So it's a little bit hard to see, but you can see some hyphae in this specimen. You'd also be able to identify yeast cells as well. No staining required for that. And then another example of, this one is a stained example, an India ink stain. This is when you would take a yeast cell from something like Cryptococcus. So this is a type of fungi. And this fungi has an enormous, huge capsule around it. That capsule is basically impenetrable. No stain is going to get through. So because it's an unstainable capsule, you have to counterstain it with something super dark like India ink, which is like this jet black stain. And then it makes the Cryptococcus cells pop out. So it's really easy to see that. Okay, let me take a break. Any questions about microscopy or gram staining or culture? Okay, 10:45 now. Let's start back up at 10:55.
I do have a question, like for like when people psoriasis come up with like What do you know more or less what area? I like German.
I do, but it was like,
But like mushrooms. But you can link it to something you might see.
I do that. Yeah. He's not gonna.
Oh. I want to use me on the wall.
With CSF, you have to be careful because there's a finite.
German finds a lot of issues.
Yep.
Yeah, that's
See something. It goes down to stain this. I saw you studying, and I was like, for example, I needed my time like, yeah, like, I'll worry about this over the weekly hack. Yep.
So, I did a run segmentation of school. And it also does what it also it does what else.
We have we're probably at
We're up the third one. No, it's the second last. The last week all we had was like.
Possible suppression is
Because after that, everything got done. Okay.
the last, yeah, the last.
The final week is we have our last test on a Wednesday and it's micro.
So we're trying to get each other. You're about down the slide.
I ain't gaslighting you.
My question that I like, what is the communication?
I don't either,
I
right?
believe his memory of
I was gaslighting you.
You
No, I think I was gaslighting myself. I think we had to change a lot. Remember me and him talking about this? We're like, okay, after this, all we have is micro left. I think I'm being gaslighted. I think we're. She must have changed it. I specifically remember that.
That's very
What? I remember.
Yeah. Very what you want to make the very more one. No.
I am the name now.
Yeah, you probably have it.
It's only like 12 hours. That's one. Is that it? That's it?
Burn all that money.
Yep.
Burn all that. Honestly, supposed to be the end. Supposed to I have to figure out what question I'm supposed to, right?
We're just talking
The weekend right before. We kind of like, hey, you have something else? Like, I'll get back to you on the 27th. That's what we do.
I said, great. Sure. So, what is the most common cause for lactic hypersecretion goes? Also known as. Oh. They're possible actors.
Yeah.
Every Friday. We work tomorrow.
Then, so you got a form that's already
Well, they were wearing like outdoor shirts.
Yeah. Major
I think that's like I'm fighting for a reason.
The only way forward is the major, the one that comes the test chair guy that came in, he's always joking around. I was like, he was wearing like the last test on Friday. He was wearing like Nike shirts. I think the faculty is there.
Yeah, they have to like tuck down.
But you never want. It doesn't say anywhere you gotta tuck it in.
Because they belt.
No, I looked at that. I hate them. So it's like it says belts have to match their shoes. It doesn't
They
say
have a belt that's not. Check one of the. Yeah. So that's
You want a cheap that got a whole ass rack and it's like you're just wearing
you
one.
bought like 10 foot amount too.
Talk about belt. What are you sucking on the belt?
You might have been.
Hey, when you understand what that's on you.
I saw some girl posting going through the
That's what I'm on.
I guess.
He keeps wanting to play Brackenridge. You know, we're gonna beat him.
So they just found a dead person.
What's that?
Breckenridge. They found a homeless man dead in the woods.
Yeah, well, he's not gonna have there make all his putts for him, so he's not taking down here. Depends on how many mulligans he decides to take.
Y'all playing this evening?
Last time we played straight up, it's like, fuck, I'm taking a drop. I'm doing a mulligan. It's like, yeah, do whatever you can do. Is this recording us right now?
They had a.
Eric, can you hear me?
That was like
It's an audio.
Look at all this nerd rage shit here.
7 of us.
Oh, it's-
How you feel this stuff?
Yeah, that was it. So you can,
I don't believe
it won't until like the day of the test might show up. I'm like, that's right.
either that or the dog's not real. Because people were saying that she and her husband were like not real people.
I feel so close to her. They're like right here. You go in the background, the split one.
Yeah.
There's a lot of like, I don't know. I think
Okay, guys, pick back up again. So next we're going to talk about a really important category of diagnostic testing. It should hopefully be fairly easy to grasp now. We just finished a block that was 60% about immunology. Now we're applying that principle of antigen-antibody binding in a way that can be of diagnostic use and valuable in the lab. So let's see how we do with some of these tests. We'll talk about ELISAs, which somebody already talked about. This is a major type of immunoassay that we use. We'll talk about lateral flow assays and agglutination assays. In general, just kind of keeping with this theme of starting out with strengths and weaknesses, the nice thing about immunoassays, there are lots of nice things, but they are pretty rapid, or they can be pretty rapid tests. And also because we're talking about antigen-antibody binding, they're generally pretty specific as well, you might expect. However, they're not always as specific as we'd like them to be, and that is because, unfortunately, sometimes antibodies don't bind exactly to the antigen we'd want them to bind to. If antibodies are binding to an incorrect antigen that we're not interested in, we call those cross-reactive antibodies. And that can be conducive to false positives, for example, so there are some problems there. But generally speaking, when you have a good, reliable monoclonal antibody that you're using, you get really good high specificity with your results. So, in terms of the types of information you can get from immunoassays, it depends, but we get good identification information. But as you can imagine, if you have an unknown bacterial specimen and you're using different antibodies to confirm that it is what you think it is, you're not going to be able to get that question answered unless you use a specific antibody for that, which means you're probably going to have a pretty good idea of what it is in the first place if you're using antibodies to detect antigens and identify the pathogen in a specimen. You can also use antigens to look for antibodies in patient serum. How might that be of interest if you're looking for antibodies in a patient?
See if they've had the disease before.
Yeah, exactly. The question you're asking in your mind might be something, have they had an acute exposure to influenza or tuberculosis or some other pathogen like that? And then what you'd be looking for, if it's acute, is probably IgM. Or you could ask, has this patient ever been exposed to, let's say, HIV? And then you'd be looking for perhaps IgG against HIV in that patient's serum. So we've talked about that a little bit in the past, but those are also types of immunoassays. An example here is specific to tuberculosis, just like I said with HIV. If you're just curious if somebody has ever been exposed to tuberculosis and they've had a chance to raise antibodies against it, you just draw serum from that patient and use Mycobacterium tuberculosis antigens to see if those antibodies are present in that patient's serum. If they're absent, then that patient has never been exposed to Mycobacterium tuberculosis. With some pathogens too, the amount of antigen from the pathogen might be present in high enough abundance where you could actually look to see if the antigen is present in that specimen as well. That isn't as commonly done, but let's say somebody does have like an active tuberculosis case. In that patient's sputum, you might be able to find tuberculosis antigen using antibodies against tuberculosis. So that is possible as well with that particular example. So in terms of the principle of this interaction, again, I hope this is pretty intuitive at this point, but this slide and this diagram is basically kind of overviewing just the principles of how we use antigen-antibody binding. In the cartoon in the bottom, or in the left, we can see some sort of plate or substrate where an antigen is bound to it. So this antigen would be something we know for sure what this antigen is, so it would be like an HIV antigen, or it would be a Legionella antigen, some specific pathogen antigen. And then the question we'd be asking is, are those antibodies in this patient's serum? That's the unknown. So we load up the patient's serum. The antibodies themselves will bind to the antigen if the antibodies are present. And then in this example here, we have some sort of detector molecule that's conjugated to the antibody. In this case, it's an enzyme called horseradish peroxidase, where if you add a substrate into that well, then that enzyme will cleave that substrate and you'll get some sort of detectable signal, an indicator that this is present in that sample, that the antibody is present. Whereas if you have a well without that antibody, you do not get cleavage of that substrate and you don't get any detectable signal. That's the principle of that assay. In terms of going back to what I was saying about cross-reactivity, this figure on the bottom right is showing some examples of how that might be possible if you use antibodies that have antigen binding sites that are capable of recognizing different antigens. You might be in a situation where you would have recognition of something that you don't want to recognize. So that would be a false positive. But most assays are very vigorously validated in QC to make sure that they are specific for what they need to be specific for and to minimize the types of antibodies that would be cross-reactive. You definitely don't want cross-reactive antibodies in those assays. Two principles we need to be familiar with, sensitivity and specificity. When we're talking about sensitivity with immunoassays, we're talking about the ability to detect very low levels of whatever we're looking for, whether it be antigen or antibody. If we want a high sensitivity assay, there's a trade-off. A high sensitivity assay means that we really want to know if it's there in the first place. So all true positives, that's what we want to know. An example that I like to use is HIV. If you suspect a patient has HIV as a screening test, you would use a high sensitivity assay like this. So you don't want anyone to be a false negative, right? You don't want anyone to actually have HIV and not be able to detect it. That's the whole goal. So you use a high sensitivity assay like this, With the risk that you might have an assay that's so sensitive you pick up some that are not actually HIV. That would be when you have a false positive. I'm sorry, false positive. So that's a risk that you're willing to accept with a high sensitivity assay like that, but that's why it's just a screening test. So you screen your patient with a high sensitivity immunoassay, you get a positive result, then you have to confirm that because you might possibly have a false positive. Yeah. Sorry, I always get confused when I'm talking about this stuff. It's super confusing.
Nice.
Yeah, sorry.
Love that.
But HIV is a good example because, again, like when you have a positive HIV test on a screening test, you never would go out and tell that patient, "Oh, I'm sorry you have HIV." That's a bad idea. You have to go and confirm that with a specific test, a highly specific test after that, so you don't give incorrect news to someone. Important tests like that. It's not urgent. You can take your time. That patient's not like they're not going to die of meningitis the next day. You can afford to do confirmatory testing and get it right so that patient receives the correct diagnosis. Okay. So high sensitivity tests. If you have a negative high sensitivity test, you should be able to rely on that patient not having that. That's the important thing, and be able to rule out that disease. So high sensitivity HIV, it's negative. You can rule that out then. That's what the point is. That's what the purpose of screening tests is. With high specificity tests, has to do more with accuracy. So this is when you would not want to use antibodies that are not super specific and highly validated to be specific. No cross-reactive binding, highly validated. Minimize false positives by distinguishing a true target from cross-reactive substances. Super important. With specificity, oftentimes with particular assays, we express this as some sort of percentage, like with malaria diagnostics. You can say it has like 95% specificity towards Plasmodium falciparum. Maybe it has a lower specificity for another type or species of Plasmodium. So that's usually how we express those. And if we have a negative on a specific test, a highly specific test, that should be a negative test. If we have a positive result on a specific test, highly specific test, that allows us to confirm that diagnosis. All these tests depend on the quality of the antigen-antibody binding interaction. You would never want to select an antibody that is against some sort of antigen that's not exposed, so it has to be something on the exterior of that pathogen, something you can actually see. And again, like all of these reagents that we use have to be carefully validated and have a lot of QC in that process. Specimen types that we would use. Serum is probably the most common specimen type, especially if we're looking for exposure in the past, so we're looking for that looking for specific antibodies in that patient. There we would draw a patient's serum, and we would have antigen-bound, known antigens. So we would apply serum to that specimen and see if there's a binding interaction. Other body fluids, anything that's in a fluid or liquid form, is usually a pretty good specimen type to use for immunoassays. Synovial fluids, urine, CSFs, things like that are useful. If we have a bacterial culture, this is a kind of a different, that's the inverse or flipped situation, where we have an antigen and we suspect the bacteria growing on this plate in this example, right? Let's say we suspect it's Streptococcus pneumoniae, and now we want to use antibodies to tell us what kind of strep pneumo it is, or what kind of strep pyogenes or strep agalactae, whatever species it is, or even group, what group those different streps are. So, like that's the flip of that, where you know what the antigen is, and now you're using antibody to detect that specimen. In terms of the generalized steps when we're talking about immunoassays, this example is showing an ELISA. Some of you guys have heard of it before. This is an ELISA where you're using known antigen and seeing whether or not the patient's serum will bind to the antigen. So in this case, we have like a 6-well plate or a 96-well plate, 24-well plate, and this plate is, like the bottom of it is coated with antigen. Then you take that patient's serum and you add it to the plate. If the patient has antibody in their serum, the antibody should bind to the antigen that is adhering to that plate. And if you rinse it away, that that antibody should remain bound to antigen. So rinsing in between these steps is really important to make sure you get rid of non-specific binding. But if it is a specific interaction between antibody and antigen, it should remain that way through these rinsing steps. So you rinse away anything non-specific, and then you add a secondary antibody. So this would be like a produced in a lab reagent antibody that's binding to the FC region, if it's there, of this patient's antibody. And then its own FC region, it has some sort of enzyme that's conjugated to it. That enzyme is what's used as a signal for the person performing the lab test as to whether or not that specimen is positive for having those antibodies. So if you have an enzyme there, just like we were talking about with horseradish peroxidase, you add a substrate. That substrate will be cleaved, and you will get some sort of indication. It could be a fluorescent signal, it could be chemiluminescent, it could be some colored product. Regardless of how that signal is received, that would be an indication on that plate. It would be something that you'd be able to read. So in terms of ELISAs, that's one thing you can do. I kept the text the same, but this is a different image on the right that's showing now instead you're looking for antigen instead of antibody. So your plate is coated with known antibody. You add antigen. It could be from like a nasopharyngeal swab from a patient you think might have COVID, for example. So let's say they have spike protein on that nasopharyngeal swab and they actually have COVID. That would be an example of an antigen that would be binding to those antibodies that are specific for COVID. After that rinsing step, if that interaction is a good interaction, you would add a secondary antibody to it, and then a third antibody that has an enzyme conjugated to it. And you add your substrate, and you get, again, some sort of signal that antigen is present in that specimen. That's a sandwich ELISA. In terms of what this actually looks like, this is an example of an ELISA and how they set up an ELISA plate. There are 8 different patients that they're testing their serum on. The way this is set up is this is a 96-well plate, so you've got 12 columns, you've got 8 rows, and then you're looking for IgG1 or IgG2b, 2 different isotypes of IgG molecules. You have some controls in columns 1 and 2 of the IgG1, and controls in 7 and 8, those columns of IgG2. And that basically gives you a concentration ladder of that antigen that you're looking for, that antigen of interest. So there's no patient information. This is all set up as like a concentration ladder, these 2 different columns. And then in the rest of these columns, 3 through 6 and 9 through 12, they set it up where you have a dilution of that patient's serum that's being added to those wells. So now we're looking, it's a little bit confusing, this setup, but this is actually a pretty typical setup in the lab. So this is one patient, S1, this is the second patient, S2, set up in these 4 wells there, and so on and so forth, all the way down. So if you look just at S1, for example, we have 4 different dilutions of that patient's serum, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000. If you still get a signal, corresponding signal, when you add some sort of substrate, you'll see a reaction, and you'll be able to read that reaction on the plate and determine quantitatively how much antigen is present in that specimen. So, and ELISA gives you an opportunity not just to ask whether or not the antigen or antibody is there, but quantitatively get that information as well through setting up dilutions like this and then reading out those results. So those are the principles of ELISAs. Another type of immunoassay that's very common is a lateral flow assay. This provides far less information. You're never going to get quantitative results with this. It's just a binary yes or no, presence or absence in your specimen. probably have all used these tests before. We rely on them a lot for different diseases and for non-diseases. This is a basic premise for pregnancy tests as well, right? Where the antigen is human choriogonadotrophic enzyme or whatever that protein is. It's in higher abundance when you're pregnant early on. So it's the same concept there. When you're using it for detecting infectious diseases, similar concept. You run your specimen, you load your specimen in some sort of port here, and then through the capillary action of that membrane, that cellulose membrane, that specimen, which is liquid, will be whipped across that entire membrane, and it will flow over some of these specific antibodies that are bound to that membrane. So if you have antigen present in your sample and it hits the appropriate antibody on that membrane, it will bind to it, and then it'll continue to get dragged across a test line and a control line. The control line is usually farther away than the test line, and that means that the specimen has to travel even farther to hit the control line. The control line is important because that tells you whether or not the test is actually working. So that's why it's set up where it's farther away. But once it hits that test line, then you should get some sort of colorimetric, usually colorimetric indication of that binding. So those are very common. Respiratory syncytial virus, influenza, rapid antigen tests, COVID, we have them for malaria, a bunch of different rapid antigen tests are available. Very nice because you have that information within a couple minutes. Very simple, anyone can do this. I didn't mean to say like PAs can do this, but like PAs can do this without any specialized training. In fact, I think you can just go down to Walgreens or CVS and pick up a lot of rapid antigen tests yourself and just put your specimen on it. So, yes, okay. Agglutination test is, I think this is the last type of immunoassay that we're talking about. With agglutination tests, we're basically drawing upon our knowledge of antigen-antibody binding and the ability of antibodies to form aggregates or clusters together. So that's where we get the term agglutination, meaning sticking together. In this example here, we have a patient's serum that is either COVID positive on the bottom or COVID negative on the top, as in they've had exposure before, so they either do or do not have antibodies against COVID. And we're just asking the question, does this person have antibodies to COVID? That's all. Okay, so if they do have antibodies to COVID, we're looking at the one at the bottom here, you add that patient's serum to some analytes. This is a commercially available, usually a latex bead that is conjugated to SARS-CoV-2 antigen. You add those together, and if antibodies are there, the antibodies will cause a lot of aggregation, visible aggregation. So you could see that on a card like this, agglutination positive results versus agglutination negative results, where you don't have any antibody in that specimen, so you get no binding. Yeah.
And are some of these quantitative as well, or is it just showing exposure at some point?
With agglutination assays, I think they're pretty binary qualitative. I don't know of any quantitative agglutination assays. If you want quantitative information, ELISAs are generally what you would use for immunoassays. But these, they're nice. You get, this image doesn't really show it, but this is a card with 6 different wells, so you can load up 6 different patient specimens on this, and within 5 minutes, usually less. Usually it's pretty much immediate. You start seeing those visible clumps of antibody-antigen clusters. So they are pretty nice for that.
And jumping back to lab workflows, like these tests, they expire. There's expiration date. What is expiring exactly? When you look at like a rapid test or a recurring test, what is expiring?
Okay, so with these, there is a biological component to these, right? You either have antigen or antibody that's involved, commercial grade, purified stuff. In this example, so you've got these little nanoparticles here, and then antibodies are conjugated to those. It's usually the biologics that determine expiration dates and shelf life. So those antibodies, they are not presumed to be good forever. And over time, what we worry about is they lose sensitivity. You have crappier binding, crappier interactions. So it's just the manufacturer will usually take that into account and know what would be an appropriate shelf life for those products, but it's because we start losing confidence that the antibodies will be able to bind and detect antibodies.
And the sensitivity goes down to those.
Yeah, if you were to find like a lateral flow assay from the 50s, which you wouldn't, because I don't think they were doing them back then, but like 70 years old,
You
you,
just taste the urine like that.
yeah,
Just
they,
pregnancy, yeah.
yep. But it would probably be unreliable for that reason. So, yeah. Yes,
So,
sir.
like, let's say if I got COVID initially, do I have to wait then for my body to start producing those antibodies?
Yeah.
And so, like, the first few days, like, I would get a negative test, and then afterwards, once those antibodies are produced, then it would produce a positive test.
Yeah, exactly. And that's a really good point. Just from this slide alone, let's say you've never had COVID before, and then yesterday you interacted with somebody who was super sick, and they were displaying signs of COVID, and they even said, "I think I have COVID." You'd be freaking out, and you could get a rapid antigen test, which would test to see if you have any antigen in your nares, nasopharyngeal swab, stuff up your nose, like all of us have had, right? But that would be for the antigen. How long would it take for the antibody to show up in your system? Minimum probably 7 days before, as a provider or even as a person who's infected, you'd start thinking, it might be reasonable for me to see if I have antibodies yet. So yeah, that was another consideration for ordering the lab test as well. Yeah. Okay, so let's have a crack at this question. What do you guys think? A, B, C, or D? B, yes, low degree of specificity. And why, without looking at the explanation on your slide?
Because it reacts to a bunch of different ones.
Say again? Yeah, so we talked about specificity being exactly like the word suggests. It's only supposed to be telling you about one thing, not multiple things. Cross-reactivity is telling you about multiple things. So, yeah. Did you have something, Scott? We'll try not to conflate those terms necessarily, because sensitivity is talking about it implies the right thing that you're looking at. You're only concerned about the right thing. So limited detection, when you're talking about sensitivity, it still means that it's a specific interaction. Specificity, you're talking about non-specific interactions. You don't care anything about the sensitivity. If it's low specificity, that's a problem that's separate. Does that make sense? Okay. Yeah. 11:20. Guess we can start getting into PCR today. How are you guys doing? Hanging in there? Did somebody say gross? Okay. You guys are okay? Push down a little bit? Okay. All right, well, it's Thursday. You have an exam tomorrow, right? You guys feeling okay about that?
No,
Oh,
not at all.
here. All right, well, yeah, let's push on with this. It shouldn't be too bad, but I say that about everything, don't I? Okay, I'm sorry, I'll stop saying that. Okay, just.
It's all bad.
Yes. Yeah. All right, we'll do a little bit of PCR then. Okay, so when we're talking about PCR, that's the most common type of nucleic acid-based test. But just know that in the, 40 or 50 years since this technology was developed, which is looking at now nucleic acids and not proteins, we have developed dozens of different offshoots of this principle, right? So PCR, and there are different types of PCR too. Qualitative PCR, quantitative PCR, real-time PCR, reverse transcription PCR, but there are also other techniques. So when we get in further to the course and we start talking about how things are detected, and we say PCR, that's kind of an umbrella term. Just know that there are other types of nucleic acid-based tests as well. And we're really only going to be talking about the principles of PCR here because it's kind of just an introduction to PCR. Okay. In terms of PCR, this is a really nice technology to have, and it is becoming better and easier all the time. If you've ever worked with the BioFire before, I think many of you probably have, that's a super easy type of PCR to do because again, you're doing minimal hands-on work with that patient specimen. If you're testing for respiratory infection, literally all you have to do is take that respiratory swab, that nasopharyngeal swab, suspend it in some sort of PDS and then load that into a BioFire cartridge. But then the rest of the work that needs to be done, extracting the nucleic acids and actually amplifying the nucleic acids if they're there, that's all done on board. So that makes it super simple. But traditionally, one of the big hang-ups with doing PCR is that you have had to do a lot of that work by yourself in a lab, which requires a lot of training and a clean environment, and then the expense of the PCR machine itself, so a thermal cycler specifically. So this is becoming more accessible and easier to do all the time. It is highly sensitive and specific, assuming that the reagents have been designed well. There are examples where you have bad primers and you don't get super specific tests, but if they're designed well, this is very sensitive and specific tests. In terms of rapidity, most clinically available PCR tests will have results between 60 to 90 minutes, depending on what type of specimen you're working with. RNA viruses take a little bit longer because they have to be reverse transcribed first, so maybe 90 minutes, but most results are very, very fast. If you compare that to overnight cultures and things like that, you can kind of see why everyone wants PCR testing done. In terms of the types of specimens that PCR can handle, it's virtually, if it has a genome, you can PCR it. And that includes RNA viruses. Anything with nucleic acids, you can PCR. So that discounts very few things. You can't do toxins, you can't do prions, but basically everything else you can do. We talked about fastidious organisms. We haven't really talked about impossible to culture organisms yet, but there are some that are pretty much impossible to culture. So that too is one of those situations where PCR is really nice because you don't have to culture anything, you just go straight to PCR. In terms of weaknesses, we talked about the requirement for specialized equipment and right personnel to run those tests. But susceptible to contamination leading to false positives, this does happen a lot, and this is why if you're running a molecular biology lab, you have to have a lot of controls in place to make sure you don't have contamination of those tests. When you're generating a lot of PCR products, what we call amplicons, they float all over the lab if you don't control them, and they can contaminate subsequent tests down the road. So contamination is a big concern for PCR. And then with primers, we'll talk about what primers are in a second, but primers are the specific part of these PCR assays. And if you don't design them well, or appropriately for the target organism that you're trying to detect, you won't get a good result. It's a garbage in, garbage out principle. So primers have to be good in order for this to work. In terms of how this assay works, again, this is based on nucleic acids. So we're looking at the presence or absence of certain specific sequences in that target organism. I could say genes, but it's not always necessarily genes. It doesn't matter if it's a gene or not. It's any nucleic acid sequence technically. In order to get at that gene, you do need to extract the DNA from that pathogen. So if you're trying to get a PCR result for a bacterial pathogen, that means you have to lyse that bacterial cell and extract out the nucleic acids and purify them first. So there's a step you have to access the DNA. Then you get your target of interest. You have in your assay, so now we're talking about a test tube with those extracted nucleic acids. You have a target, you have a probe that is specific for that target, and that probe is nucleic acids itself. They're called oligos or oligonucleotides. And that probe is conjugated to some sort of signal again that gives the user an indication of whether or not that pathogen is present in the sample. The vast majority of them these days are going to be fluorescent signals, especially in clinical labs. So there are detectors on those PCR platforms that give you an indication of whether or not the specimen is there, fluorescence detectors. In terms of how this actually works, in order to amplify DNA from a specimen to the point where you have a detectable signal, you have to be able to polymerize or synthesize additional copies of that target strand. In order to synthesize new copies, we take advantage of the fact that we can use temperature to either dissociate double-stranded DNA or bring it back to rehybridize to itself. So that's why we use platforms called thermal cyclers, so we can control the temperature and the length of time those specimens are held at respective temperatures, and then control the amplification process. So we start off with our double-stranded DNA. We will crank up the temperature to a really high temperature, about 95 degrees centigrade, just below boiling. We'll keep it there for about 30 seconds, which is just long enough to make sure that those hydrogen bonds that keep the double-stranded DNA in place are broken. But that's a temporary thing, right? So we heat up to 100 degrees, break those hydrogen bonds, and now we have our two separate strands that we can amplify if we have a polymerase present in that tube. So we have to have a polymerase as well, and a bunch of nucleotides. If our target sequence is present in that specimen, our primers that we have, which are also small little nucleotides, will bind to specific sequences on that target template. And they will have to bind specifically to both strands. So you have a top strand here and a bottom strand. We already know that polymerases only synthesize 5' to 3', right? We learned that. So that top reverse primer is going to be synthesized in one direction. It's going to be 5' to 3'. And then that bottom primer is going to be also synthesized 5' to 3'. Okay. The end result, if we have that polymerization working, is that you have second strand now on both of those parent strands. So we've essentially doubled the amount, our target DNA specifically in that region. That's one cycle. So we're controlling this by temperature. We've gone from having one copy at 0 cycles, and now we have 2 copies at 1 cycle in. If we continue this cycles for another, for a third cycle, I'm sorry, a second cycle and a third cycle, we'll continue doubling the amount of DNA present in our specimen. And these cycles are happening pretty fast. So it takes maybe 30 seconds to denature DNA, a little bit longer to anneal primers to that target DNA, and then you have to allow for a little bit of extra time for extension to happen, which is the polymerization, or the synthesis of that other strand of DNA. But that's what goes on with each and every cycle. So that by the time we run through, let's say, 24 cycles of that, exponentially we have 16 million theoretical copies of DNA where we just started initially with 1. Okay. And that is certainly detectable, or it starts to get into the range of detection. In terms of what that looks like, this is kind of like a typical result from doing a quantitative PCR on multiple patient specimens. So each one of these curves represents a different patient specimen. And on the x-axis here, we see the number of cycles that specimen has undergone. So the more cycles you see, that means you should have more DNA present. You've amplified all of that if it's there in the first place, if the target is there in the first place. If the target is not there, you will have a flat line. It means that there was never any amplification because the primers never had anything to anneal to in the first place. So that's a negative result. But for the positive results, eventually, if they have fluorescence that's being released during these amplification cycles, that fluorescence reaches some critical threshold value, and you start getting this exponential curve because you have exponential amounts of target present. And since this is done on like a PCR machine, most of them have software that will sit there and analyze those curves and tell you when a sample becomes positive. But you as a user can also visually see this and say, you can watch it real time each and every single cycle as it starts amplifying on those positives and see when those positives start coming up. Okay. that makes sense. And you also always have controls as well, so you're not just running your PCR without controls. You put a known positive on that run, and that known positive has to be amplified. You also put a negative control on there, usually water or saline or something like that, that has no target. And if that amplifies that negative control, no template control, then you have contamination, and you've done something wrong, and your assays needs to be redone. Okay, so we can use this to kind of back calculate relative amounts of concentration of different pathogens based on when these amplification curves meet a critical threshold. That's called the cycle threshold, or CT value. When you're doing quantitative PCRs, qPCRs, that is oftentimes an important thing that you're recording, which CT value those specimens became positive at. And this becomes important for a couple different types of specimens I'll talk about in just a second. A couple other features to note here. In terms of the entire time it takes for specimen processing and the test itself, is usually between 30 to 75 minutes. If you're doing sample processing, it could be a little bit longer than that. Extraction takes some time as well. But you do not need a lot of material. Sample lots are typically very small, and that's because you really only theoretically need a single copy to start with in order to get a positive result. In practice, usually you have quite a bit more than a single copy to start with, but that's theoretically what you need. What does the word aliquot mean?
Specifies.
Yeah, a portion of a specimen. So if you draw from a lumbar puncture, I don't know, like 300 microliters of CSF from a patient, you're not going to use all 300 microliters of that precious cerebrospinal fluid for one test. You're going to pull off a small amount, a small aliquot, a small portion, and give it to the molecular lab to run their PCR, and give it to another portion of the lab to do another diagnostic test. But that's what aliquots are. In terms of limitations, it has a lot to do, there are a lot of things that can go wrong with PCR, especially if you're doing like full complexity PCR, not the simplified gene experts and biofires. It is possible that you do not meet that threshold, like there's not enough starting material, or your conditions aren't right to amplify it, but it's there, and you can get false negatives. Take, for example, if we look at this curve and we're talking about, let's talk about COVID. So you've got presence or absence of this virus. There's going to be a point in that patient's infection where they have too little present to be detectable. That's the limit of detection that applies for PCR as well. It could be at the beginning of their infection, or it could be at the end of their infection, but it is possible. The good thing about that, though, is if there's no detectable amount in the specimen, they probably are clinically going to be fine in the first place. Okay? Another potential problem are false positives when you have nonspecific binding with those primers or with the probes. But again, that's pretty uncommon, especially in clinical settings, because you don't design primers that don't work. In order to have a clinically validated, like FDA-approved PCR assay, those primers and probes have to go through really rigorous testing and quality control. So they should work, they should bind where they need to bind, and you shouldn't have any problems with false positives. Research labs have a little bit more latitude, so it's more common for us to have false positives, but the only other thing to be aware of, or the fact, with at least RNA viruses, which was what I was talking about with SARS-CoV-2, they do mutate naturally, right? So over time, the sequences that we use to design those primers and probes off of, they do mutate. So eventually, those assays may not work, but again, like we keep doing a lot of surveillance work, we keep sequencing, circulating isolates, and we know what those sequences are and if they need to be updated, like the primers and probes, they need to be updated. Okay, in terms of, this is another really, really important slide. I think it's the last one on PCR, but super important stuff is going back to how PCR can be used quantitatively and how that's good for providers to use to guide patient therapy, clinical therapy. if you have a patient, particularly with one of these viral infections, like an HIV positive patient or a patient with not just any herpes infection, but let's say you have aseptic meningitis, a really critically ill patient who has herpes infections, or somebody who has hepatitis B, those types of infections. Once you diagnose that patient as having one of those viral infections, you will treat them for sure. All of these different viral infections require treatment. When you start treating them as a provider, you want to have a way of monitoring treatment efficacy. So, you put them on acyclovir if they have herpes, you put them on some antiretroviral for HIV, and then you need to know whether or not that is impacting their viral load. So you can run their serum samples over different time periods and see if that is diminishing the amount of viral DNA or RNA in that sample. Very, very useful for those things. You can also use those assays for looking at antiviral resistance and other trends over time. But the way that works is, this is a contrived example, but it shows you quantitatively how you would see those curves coming up at different cycles. This is a known concentration ladder. So on the left here, this assay has been set up where you have decreasing amounts of input DNA into this assay, tenfold decrements from 1 microgram all the way down to 100 femtogram. So that's 1, 2, 3, 4, 5, 6, 7, sevenfold decrease in the amount of starting material that you input into this assay. You can use those cycle thresholds that come up for each one of these known volumes, and then you can run your unknown patient specimen, so your guy with HIV or whatever, and say his CT comes up right, I don't know, like right here at cycle 23, he starts amplifying there. You can back calculate what his viral load would be based off that known standard. You don't even have to do that, the software will do it for you. So, yeah. Does that make sense? Okay, cool. Let's just power through these last couple slides, okay? You guys look like you're definitely losing me. I'm sorry, but it would be great to pick up on antibiotic susceptibility testing tomorrow. So this should be, I'm not even going to say it. All right, so we've been mostly talking about methods to detect bacteria, with the exception of that quantitative PCR slide that we just finished on. But real quick, with viruses, it does involve a completely different approach because we're not going to be able to culture them. We're not going to be able to gram stain them. If we have like a MALDI or something like that, very limited use for MALDI-TOF and mass spectrometry. But what we do rely on for patients with suspected viral infections would be rapid antigen tests, which we have a ton of rapid antigen tests for viral illnesses. We've got direct fluorescent antibody tests, DFAs. You need a fluorescent microscope for that too, but it's a really simple, quick test you can do if you suspect a patient has viral illness. Electron microscopy would be great if you had the facility to do it. Look at this wonderful micrograph. The virus is budding from a cell. PCR is probably the most relied upon diagnostic test for viral illness. So, and as I said, it doesn't matter if it's an RNA virus or a DNA virus. With an RNA virus, we just synthesize a second strand for DNA and still run our PCR. Serology is also important, but when we're talking about antibody detection, remember that that's not, I mean, it's still an immunoassay, but that is different than the rapid antigen tests. In serological testing, we want to know whether or not that patient has been exposed to herpes or HIV or whatever. Okay, so that is a slightly different interpretation for different diagnostic purpose. And then, as I said before, in block 1, we can do viral culture using our phase contrast microscopes in labs that some of them still have, where we would read out whether or not the infected susceptible cell lines are capable of being infected and have cytopathologic effects, CPE. So those are kind of the mainstays, particularly PCR and serology and rapid antigen tests. For fungal pathogens, similar to bacteriology, but a little bit different. Microscopy is still useful, but there's no peptidoglycan, so we're not going to do any gram stains. KOH preps are important to be able to do for fungal specimens. A number of other types of specialized stains for fungal specimens are useful. Gross colony morphology for culturing fungal molds is important. It takes a very, very long time though to grow up molds relative to yeast and relative to bacteria. So there's a little bit of a drawback there. There are a limited number of biochemical tests that we can use for fungal pathogens. I don't know how much labs rely on biochemical tests, but it is possible. They do have their own distinctive profiles as well. Molecular methods are fairly common for fungal pathogens. We can still do PCRs on fungal pathogens as well. This is not on the list, but we have some antigen tests for things like Cryptococcus is one where we use antigen testing for diagnosing Cryptococcus. Oh, sorry, immunoassays, yeah. So that would be an example of an immunoassay. It's fungal antigens. And then for antimicrobial susceptibility testing for fungi, that can also be really important depending on the fungal pathogen. So those are usually pretty involved types of AST. They're a little bit different than how we do it for bacterial specimens. Oftentimes clinical labs will send out fungal specimens for reference testing. Okay, last one. Talking about those new, faster technologies. Some of you may have heard of these, but they are becoming very, very common in DoD labs. Common enough where you might want to be familiar with them. MALDI-TOF. You guys, anybody heard of MALDI-TOF before? A couple of you. This is a mass spec platform here. It stands for Matrix Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry.
Nice.
Yeah, it's a lot of words, but the nice thing about that acronym is it walks you through exactly what's happening. But yeah, basically, like it's mostly validated clinically. Most useful right now is still for identifying bacteria. It just identifies them based on mass to charge ratios. So you put your little bacterial isolate, your pure colony associated with some matrix, some analyte. Put it on a little stainless steel dish and load it onto that instrument at the bottom here. And then once you load it into that specimen port, a laser zaps that specimen, basically ionizes it, it desorbs, and it gets like shuttled through this entire vacuum tube right here. That's the time of flight portion of it, and ionized by that laser in the process. So whether it's all of those different peptides that are ionized in the process have different unique masses and different charges, so their mass to charge ratio matters when they hit this detector plate that's on the far end of that vacuum tube. And that detector plate will convert those signals into a profile that can be read out as a spectrum and then associated with different types of bacteria. So, the longer this tube is, the better the resolution and the higher the accuracy of the results, but generally these are pretty accurate machines, and you get results instead of hours, instead of days, you get results within 30 minutes, so super fast. What else? Sequencing technologies. This is a MiSeq. It's a fairly high throughput platform where you again do an extraction and prepare the genomic contents of some target of interest, and then you sequence the genome and then blast it against some database to see if it's positive for that particular pathogen, see if it matches with known pathogens. And this is another sequencer. This is a tiny handheld one called MinION from Oxford Nanopore Technologies. So the DoD is starting to use now. MinION's more, and we've been using MiSeqs for some time. Okay, we'll call it a day. You don't have to know how this stuff works, but just know that these are platforms that when, like, particularly in MALDI-TOF, maybe not so much sequencing, because clinically that's hard to validate those. But with MALDI-TOF, like some of your patient results are going to be coming off those instruments, so I wanted you to have some idea of how they work. Okay. Yeah. So tomorrow we'll start talking about antibiotic susceptibility testing, and then we'll. All right. Sorry about that, guys. Have a good test tomorrow. Good luck.